This is slide 7, our 4 nucleate stage. We are again looking at the ovary. It's again a cross section through our ovary with the pointer currently on an ovule within the ovary. So that is the first term that you need to identify, an ovule. It's the same as in our last slide. Here's one, here is another. There are 6 total seen in this slide. So we're going to go back to this one. We are at the lowest magnification at this point, but you can see that surrounding the ovule there are these layers of cells that are called the integuments. These are layers of cells which surround the developing megasporocyte or megaspore, and they will ultimately produce a seed coat. The seed for a plant, an angiosperm, develops from the ovule. The outermost parts of that will form the seed coat. These are the layers of cells that are called the integuments here. Now that's about all we can see at this magnification, so I'm going to increase to about 100 times. We are looking at an ovule here, and now you can start to see that there are distinct bodies inside the where we had previously seen the megasporocyte. As I mentioned in the last slide, the megasporocyte underwent meiosis, but only one of the 4 daughter cells from that mitotic event survived, and that was called the megaspore. Now, the megaspore undergoes 3 successive mitotic divisions. So we are going to ultimately see that one cell, the megaspore, divides into 2, then into 4, then into 8 separate structures. Now, I have to also mention that although there is division of the nucleus, there is not a cytoplasmic division between these offspring from the megaspore. So you're going to have an 8 nucleate cell eventually. Once we fully mature and get to the mature gametophyte, it will be an 8 nucleate structure called the embryo sac. So at this point, what you can actually see, we will zoom in once more, but you can see that there are 4, 1, 2, 3, and 4 structures within this ovule. So we will increase our magnification once more to about 400 times so we can see that a little bit clearer. So again, here is what had been our megaspore. It has undergone its first 2 mitotic events, producing 1, 2, let me focus on that a little bit, 2, 3, and 4 nuclei in this one space here. So this will ultimately become an 8 nucleate mature female gametophyte, and we will look at that on our next slide. But right now we have the ovule, which is this entire structure here. And within that, we have our developing embryo sac here. It's currently at the 4 nucleate stage. The integuments, again, were the layers that surround the developing embryo sac. So right here, there are layers of cells on either side, here as well. These are the integuments. They will become the seed coat. And the last term that we are asked to identify is the micropyle. That is right here. As you can see, there's a slight space, a division here, which is a pore that will allow the sperm, remember our sperm is currently traveling down our tube cell, and it will enter to fertilize the egg, which is going to eventually develop in here. And so the sperm have to enter somewhere. The tube cell will terminate here at the micropyle and allow for the sperm to enter this space to access our egg. So I'm going to just show you that in another ovule as well, just so you can see it a little bit better here. Here's another ovule with the integuments here. And as you can see, there's a space here. That space is the micropyle. Okay, right here. Okay. All right, so that's it for slide 7. We will continue looking at the mature female gametophyte at the 8 nucleate stage on slide 8.